Sharon Courtney, and we're here to talk to Dr. Jay Rappaport, the director of the Tulane National Primate Research Center, where they're working to make vaccines safer and more effective. Has the Tulane National Primate Research Center, or TNPRC, done research to combat viruses and infectious diseases before COVID-19?
Our research, about 65% of it is focused on HIV, AIDS. We do vaccine work. We've contributed to many of the drugs that are currently, the development of the drugs that are currently used in therapeutics for AIDS. We have other avenues of research where we work on Lyme disease. The test for Lyme in humans and animals was developed at TNPRC. We're developing newer diagnostics and treatments for Lyme and even post-treatment Lyme disease. We're doing research on aging, Alzheimer's disease. We're really expanding our portfolio into many different areas. We've done work on Zika virus in the past, other pathogens. Our major strength is infectious disease at the Primate Center. And we're the only national primate center that has a regional biocontainment laboratory. This biocontainment laboratory is at the level that is needed for work with the COVID-19 virus. We have the largest capacity at this level, at what's called biosafety level 3 in the country, and we're highly suited to do this sort of work. We have aerobiology, which we can really study what the particle size that carries the virus. We can see if it's stopped by masks or if you need a respirator. We can determine if the sequence changes before and after infection by looking at the RNAs or proteins within the infected animals or the infected cells. We can measure immune responses and determine which ones are protective and which ones may be harmful. There are so many things that we have the capabilities to do. And I think that's the work we can do, I think would most rapidly move the field forward in terms of generating therapeutics that work, vaccines that were effective, and diagnostics that could best pick up infected individuals and prevent the spread.
We all hear that the TNPRC is a tremendous asset, especially when combating infectious diseases. Why?
Well, actually, we were founded, to combat infectious disease. That's really our major strength. We have enormous strengths in immunology, virology, and in terms of our capabilities, of the 7 national primate centers, we are the only one that has a regional biocontainment laboratory at biosafety level 3. So we can actually work with pathogens that are dangerous, and we have the largest capacity actually in the country, possibly in the world.
So how is a non-human primate model better than other animal models?
The immune system in non-human primates is very similar to that in humans, and we've seen that in our AIDS vaccine studies and a number of other studies. There's been many studies that have been done in mice that don't translate into work in humans. In other words, there are many drugs that have come out of, or even vaccines that have come out of mouse studies that prove they're not effective when you move into a non-human primate or human model. So the non-human primate model is the best, and will likely save lots of time in terms of finding the right vaccine that's going to work and the right vaccine to then test and then ultimately deploy into human beings.
So we hear that people like those over 60 are more likely to be at risk. Is the TNPRC focused on any special risk groups?
Our initial studies on the animal model are actually focused in older animals. For that reason, we want to have the best model that best recapitulates what goes on in human beings. So our initial focus will be the aged animals. We have a colony of rhesus macaques that's supported by the National Institute of Aging. So we have animals at many different ages. And so we're going to be using some of these animals. And we also have African green monkeys where we're going to use some older animals too for the same reason. And then we can compare young versus old and really see what the effective aging is.
Will there be a non-human primate model for COVID-19?
It looks like in the rhesus model that it looks like the infection doesn't cause a really severe disease, but we're hoping that the African green monkeys are better and give that more severe pathology that we're looking for. At the same time, we're planning strategies that will enhance the disease in the rhesus or the African green. We have several strategies planned to create the best model possible.
Other than the model, does the TNPRC have any other kind of research that can be useful for COVID-19?
We're doing studies on therapeutics, diagnostics, and vaccines in those three different categories. We have multiple approaches that are planned. There are RNA-based approaches, protein-based approaches. It looks for vaccines that, because antibodies appear to be protective, that an RNA-based approach or protein-based approach could be highly effective as a vaccine. There are multiple companies now that are lined up with therapeutics that they want to test with us. And also there are investigators here at Tulane that have strategies that they want to test in vitro in the primate model. So yeah, we're really working at multiple avenues. Tony Hu, Dr. Hu, has been working with the Primate Center to develop a highly sensitive test for COVID-19 from plasma.
There's a lot of concern nationally about the use of animals in research. Can you talk a little bit about that and talk about how the animals are cared for?
Okay, this is extremely important to develop treatments and diagnostics, vaccines for infectious diseases. This particular infection with this coronavirus, I think, underscores the need to use non-human primates and animal models. We have the best animal care available in the country. So we're really, we treat the animals extremely well, and we care for them, and we care about them.
This isn't the last time you'll hear from us, and we want to know what questions you have. So please email us at medquestions@tulane.edu.